Today's focus, as Michelle mentioned, is really looking at matching patients to cancer clinical trials. But we hope with this combined series of talks that you'll walk away with a more fulsome understanding of the current trial environment in Canada, matching patients to precision medicine trials, which is an increasingly complex trial finding, trial matching process due in large part to the added considerations for patient eligibility that come with those trials. Over the series of presentations this afternoon, we will learn a little bit through from different perspectives, starting with Dr. Enrique Sanz Garcia's presentation, where we'll look at the elements of trial design and the value in matching patients in precision medicine trials to improve their outcomes. And we'll move into, with my talk, an overview of the trial ecosystem and the current active trial environment and how patients and caregivers and researchers contribute to that landscape, but also how patients and caregivers navigate finding trials, which may be surprising for some, not a straightforward process, and then there are some inherent limitations still in the system. From there, we'll pivot to, and I'll also talk about some resources available and some best-in-class resources available supported by 3CTN for finding and navigating the process for patient navigation, rather, the process for finding trials. And then lastly, that will set the stage for Paul Brogee's talk on automated matching and really what can be representative of the future of trial matching of cancer patients to trials, highlighting the Ontario-based PMATCH project, for which 3CTN serves as our network of 51 cancer centers serve as a receptor group for this project when it's completed and ready to be applied to the clinic. So look forward to hearing from Paul more about the PMATCH and how that proposes to improve and really move forward and improve the efficiency and effectiveness of the trial matching process. So that's what we have outlined for everyone this afternoon. Hopefully, there will be some questions that you will have as a result of these presentations. But before I introduce Enrique as our first speaker, I'll provide a little bit of a background on Enrique Sanz Garcia. He's a medical oncologist and staff physician at Princess Margaret, with a clinical focus on head and neck and gastrointestinal cancers. He completed his medical degree in Spain, as well as a PhD in medicine, focusing on liquid biopsy and colorectal cancer. In 2020, he moved to Toronto for a fellowship in drug development in the head and neck program. And following his fellowship, he was appointed as a staff physician at UHN. His main areas of research include precision medicine based on medical profiling, liquid biopsy, and early onset cancers of the GI tract. He serves as the scientific co-lead for the Marathon of Hope Cancer Center program at Princess Margaret, chairs the molecular tumor board for this initiative, and co-chairs the working group for the development of a prospective study to bring precision medicine to cancer patients across Canada. He has been granted a Young Investigator Award from the Conquer Cancer Foundation of ASCO, as well as a Merit Award from ASCO and the Novartis Oncology Young Canadian Investigator Award for some of his research.
Thank you so much for the introduction, and thank you so much for inviting me to this meeting. Perfect. So in the next 20 minutes, I would like to speak a little bit more about how we are trying to match patients to clinical trials based on molecular findings. As I mentioned, it was mentioned before, I am a medical oncologist, and I am a clinician investigator, which has a special interest on precision medicine and improving the efficiency of clinical trials in cancer patients. So, as you all are very aware, medical oncology is a field in which precision medicine has had a profound impact over the last few years, and we have moved from the previous paradigm of one size fits all to a more personalized strategy, trying to find the most suitable treatment for every patient, not only based on the tumor type, but also on the personal characteristics of the patient and the molecular biology of the tumor. This has been made possible thanks to the introduction of new drugs into the pipeline of development and also into the clinic, like molecular target agents as well as immunotherapy. The clinical trial design currently is moving more into a more patient-centric and inclusive approach, although, as I will say later, there is still room to improve. And we are trying to optimize outcomes in those trials by integrating biomarkers into the eligibility criteria of patients into those studies. However, we have still significant limitations, which I will also highlight in the next few minutes, regarding the access to clinical trials across the country, as well as the access to the diagnostic tests that are needed to identify those biomarkers that could open potential opportunities for patients into those clinical trials. When you look into the most recent data of actionable alterations based on OncoKIB, you can see that in the last couple of years, there has been a lot of now new indications and new biomarkers that have been associated with predictive response and an FDA-approved drug. For example, you can see that we have gone from 45 to 55 genes. And similarly, there has been other biomarkers that have been recommended by different guidelines, and they are following a potential approval by the FDA, moving from 23 to 31 genes. This also shows that we have more rationale to include patients on clinical trials because there are more research and more clinical evidence that some biomarkers can be useful. And as you can see also here in this graph, in the last few years, there has been some biomarkers using the OncoKIB therapeutic levels of evidence that have been granted for further investigation. When we look into the alterations that in solid tumors we tend to look at, we are looking mainly to mutations, fusions, translocations, as well as gene amplifications, at least from the genomic perspective. The most majority of the approvals have been related to specific tumor types, like for example, lung cancer or colorectal cancer. They have their own specific tumor approvals based on molecular alterations. However, in the last few years, there has been a few drugs and a few biomarkers that have been approved based to any kind of solid tumor, following what we are called a tissue agnostic approval. Some examples are shown in this table. For example, NTRK inhibitors for NTRK gene fusions, including ralutrectinib, entrectinib, or repotrectinib, BRAF V600 mutation in non-colorectal cancer with dabrafenib and trametinib, RET fusions with serpercatinib, NRG1 fusions with zenocutuzumab, as well as high expression of the surface of HER2 following histoimmunohistochemistry with thadinatamab. Those, as I mentioned, are tissue agnostic approvals. Also, some of these tissue agnostic approvals have been associated with response to immunotherapy, like for example, deficiency mismatch repair or MSI high status for pembrolizumab and dostarlimab, as well as high tumor mutation burden for pembrolizumab. When we look more specifically about the different clinical trials that are currently ongoing using precision medicine and trying to maximize outcomes and potential enrollment of patients, I want to highlight that there are three main designs. The basket trial, which I will explain later, is usually associated with different tumor types that they share the same abnormality, usually a mutation or a fusion, and then the patients are treated with a specific intervention. The second option is an umbrella trial. That's something that is associated with one specific tumor type, and every patient could receive a potential treatment based on alteration that has been seen in that tumor. And the most sophisticated approach, which I think is the one that many companies and many cooperative groups are moving for, which is a platform study, meaning that patients with specifically with one tumor type, they are assigned to different treatments based on characteristics of the tumor and molecular biology, and every arm is independent to each one. So when that drug has some efficacy, is moved forward, and when there is no efficacy, they can open all the different arms, meaning that in one single study, we may be able to test many different drugs in many different patients in a specific indication. This table is able to summarize the 3 different clinical trials that I think are running right now in terms of precision medicine. Basket trials have been the most classic ones, and I will explain that a little bit later. Some examples are NCI-MATCH, My Pathway, or Aurora study that has led to the approval of BRAF inhibitors in patients with BRAF V600E mutation. Umbrella studies are mainly on lung cancer, although there are other tumor types, like they are also trying to get those studies moving forward. And finally, platform studies, which as I mentioned are the most innovative one because they are very flexible. They can evaluate and optimize multiple therapies over time, and they have proof to accelerate the drug development and real-time adaptation. When we look into precision medicine basket trials, and this is a table that has been shared with me by Dr. Lilian Su, you can see that there are 3 main studies that have been so far published or are under development. The largest one being the NCI-MATCH study, which I will explain later. I want to highlight a couple of things about this table. The first thing is that I want to highlight is that despite we are testing many patients, most of the studies more than 2,000 patients, you can see that only half of the patients in TAPUR study and almost 2/3 in DRAP study, they have actionable alterations, but only half of the patients, even lower, 20% in MATCH study, they are able to have a complete match to a drug and be able to benefit from a potential therapy. Moreover, when we look to response rates, which are the main endpoint of these kinds of studies, you can see that the response rate has been ranging from 0% in many arms to up to 46%, for example, in BRAF inhibitors, meaning that despite we are able to test many of these patients, only a few patients can have a match, and from those patients, the benefit of the drugs are also quite small in many of the arms, and overall only 23 or 34%, as you can see in these 2 studies. As I mentioned, the larger study is the NCI-MATCH study. This is a study that is currently recruited and has recruited more than 6,000 different patients. Only 1,500 were assigned to a treatment arm, and this table that was published a couple of years ago, it includes only 37 studies, but there are up to 45 studies at this point. And from those studies, only 7 studies met the primary endpoint, showing enough response to try to move forward for a later phase of development. I want to highlight also that the majority of these studies, they have 6 months progression-free survival, which was below 50%, meaning that we have still room to improve to select those patients and also to bring precision medicine alternative to these patients in earlier stages. However, one of the learnings of this study is that we could see some meaningful response rates in strange alterations, like, for example, ALK or FGFR mutations, and also in red tumors, bringing the opportunity for precision medicine for those tumors who are not as common as colorectal, lung, or breast cancer, and they have less access to trials or potential therapies. This is an example also of an initiative here across Canada. This is the CAPTURE study that has been led by the cooperative group of the Canadian Clinical Trials Group. And this is a study that is currently recruiting in some of the arms. This has been an example of how in Canada we can also do precision medicine using clinical trials. There has been some struggles in terms of accessing drugs, although thanks to the CTG that has been easier. And the other main issue has been access to testing those alterations in patients, which I will show you later is also a struggle in the real practice at this point. So once I introduce a bit more about what has been done so far in terms of precision medicine and what are some examples of the clinical trials that we are currently running, I want to focus the next few minutes of my talk to talk more about the next steps in designing clinical trials and what are the current limitations of matching patients to clinical trials based on biomarkers. I will start first by saying that across Ontario, we have a problem in terms of access to genomic testing through a standard of care, because next generation sequencing that has been so far the gold standard has been only covered for certain cancers, like for example, lung cancer and thyroid cancers. And we know that we have many other tumor types that could benefit from this approach. Another limitation for matching patients to clinical trials has been that many of the biomarkers that we are currently running on our research portfolios, using, for example, whole genome transcriptome sequencing, they are not yet well understood, they are rare, and they are not fully validated, meaning that to move into a clinical trial perspective, we may have to get some improvement on that prior to be able to get access patients into those trials. Another limitation on clinical trials has been the geographic, socioeconomic, and racial disparities that has been affecting patient inclusion, and that has limited the conclusion of some of the studies that have been published in the literature. One other limitation, which I think Paul is going to try to overcome in his presentation, is to try to improve the integration of medical records, genomic data, and clinical trial database to make sure that every patient gets at least a match for a clinical trial. One limitation that is also a matter of education through the community is the low awareness of precision medicine trials among clinicians, among patients. We receive many self-referrals of patients who are aware about potential precision medicine initiatives, but we also want clinicians to refer patients to academic centers to try to improve the recruitment on these kind of trials. And finally, but last but not least, I would like to say that many of the clinical trials are quite restrictive in terms of eligibility criteria, especially when we are looking into phase 2 and phase 3 studies. And these are excluding patients who may really benefit from these drugs, and they are not performing real-world patients studies. So I think that's something that is getting improved in the last few years, but I think it's also a limitation for improving the efficiency and the conclusions on clinical trials. So as I mentioned before, I think the next step is to move beyond the next generation sequencing panels and looking beyond the DNA, and more specifically, moving beyond the hotspots testing. And one of the things that has been tested in the past, and we are trying also to work here in Princess Margaret with the help of the OICR, is to try to evaluate the role of transcriptomics to predict response to therapies. And this is something that has been shown by different studies over the last few years. This is a study led by the WIN Consortium back in 2029. And I want to highlight from this study that they were testing DNA and RNA in patients who have advanced tumors. However, the truth is that the RNA arm was only evaluated when there was not any alteration in the DNA. But as you can see here, 22% of the patients who were consented were able to get a drug based on matching of the DNA, while 12.5% were able to get a drug based on the RNA results. And when you see here, you can see how the clinical benefit, which was measured by stable disease more than 6 months, partial or complete response, was slightly higher in patients who were matched to a drug based on RNA, bringing the point that I was going to make, that probably matching patients to RNA results is also worth to explore. As I mentioned before, this study has a limitation because only those patients who fail for DNA matching were selected for RNA sequencing and therefore matched to a therapy based on the RNA. But our colleagues at BC, they have done a nice work to show that RNA sequencing is important and that whole genome transcriptome sequencing can improve the matching of patients to drugs and clinical trials. This is the POC study that you may all be aware about. You see here that for a total of 700 patients at that point of publication, 80% were able to undergo successfully a whole genome transcriptome sequencing, while 66% of the total were able to get an actionable alteration at the molecular tumor board after discussion. From the total of patients, only 29% of the patients were able to be treated. Many of them, they failed because they were too sick to get into another line of therapy. And despite the number of patients who get a clinical benefit from the total was only 16%. That there was a clinical benefit among those patients who have an axonal alteration of 46%. And more interestingly, the part of the WTS that contributed most to match patients to a trial or to a potential therapy was the RNA, bringing the point that I was making before, that looking beyond genome is also a way to go to try to improve matching patients into clinical trials in precision medicine. For that purpose, I think the Marathon of Hope Cancer Center Network is making a huge effort to try to bring precision medicine to patients. And one of the main hopes and goals in the last few years has been to do a whole genome transcriptomic sequencing in patients on real time in prospective ways, so we can get some alteration in those patients, and we are able to match them to potential clinical trials. And this is something that here in Ontario we are doing, thanks to the Marathon of Hope Ontario protocol that we are currently performing at Princess Margaret, and is hopefully opening soon in Ottawa and also in Windsor. So this study is focusing at this point mainly on solid tumors, although we are trying to validate this also in hematological malignancies. And in partnership with OICR, we are able to perform in a timely manner a whole genome transcriptomic sequencing in patients who are currently under treatment with a clear certified report. The only caveat about this study is that we need patients who are able to wait 2 to 3 months to get all the results, because from the moment that the patient consents until we get the results, there is a lot of work that is being done behind the scenes. And we are currently using both archival and fresh tissues. We know that fresh tissues are better to get results for whole genome transcriptomic sequencing, but thanks to the OICR team, we have been able to develop a quality control check early on the analysis of the archival tissues. So if they are not good enough for WTS, they can be transferred back to Princess Margaret for next generation sequencing. Once the results are back, we have a molecular tumor board that is meeting right now every week, when we have clinicians, scientists, genomics, geneticians, and we discuss the clinical background of the patient, the main alterations, and then we have a literature review about the potential therapeutic options, and also we try to have representation from clinical trials unit to try to match patients to potential therapeutic options. A couple of examples from our tumor board in the last few months. This is a case of a patient with thymoma, which is a rare cancer. This case only shows a mutation in that gene TNFAP3, which has been associated with autoimmune diseases. And as you may know, thymoma is a tumor that is related to the thymus, which is an immune organ. But the most important alteration for us was this finding of the RAD50 alteration, especially a deletion that has been associated with potential sensitivity to PAP inhibitors and also potential benefit of radiation. So we are hoping that this patient in the future can be matched to trials evaluating PARP inhibitors or drugs, including platinum chemotherapies. Another example is also this patient with a mesothelioma. This patient had an alteration on MTAP, especially with a deletion. This is something that in the past we were not looking at, but thanks to the WTS, we are seeing that quite often in our reports. And this is a very interesting alteration because there are many drugs, as you can see here in this table, that are currently under development targeting that alteration. And we have some clinical evidence in phase 1/2 studies that shows some preliminary efficacy in patients with this deletion, as well as reduction of the tumor in the majority of the patients. So finally, I would like to use my last minute to discuss briefly how we see the clinical trials moving forward in the future. Our goal or hope that we all share is that we may be able to characterize at baseline the tumor and tumor microenvironment. And then, thanks to the development of machine learning, artificial intelligence, together with the clinical and non-clinical knowledge of different anti-cancer agents, as well as the experience that we are getting from other patients, we can really fit every patient into a clinical trial or a precision medicine strategy. And even more, once the patient is in those strategies or in that clinical trial, we are able to monitor the patient carefully during the treatment with different liquid biopsy and dynamic biomarkers and see whether there will be a potential benefit or not, and switch patients quickly prior to a radiological progression. So this is my last slide. That's to conclude. As I mentioned before, I think biomarker-based patient matching improves trial efficiency and outcomes. And I think the platform studies that are currently ongoing are a good way to maximize efforts in trying to recruit patients in trials, as well as to making patients eligible for getting into trials. I think one of the main things that we need to advocate is to improve the access to biomarker testing. We have this wonderful initiative from Terry Fox and Marathon of Hope to do whole genome transcriptome sequencing, but I'm also looking forward to see other proteomics and immunohistochemistry to try to include, for example, antibody drug conjugates, which I didn't bring up in my presentation today. I also believe that including patients with a broader inclusion criteria is good because it may reflect better real-world populations. And as I mentioned in my last slide, I think that integrating molecular data, clinical characteristics, as well as artificial intelligence can enhance patient matching, boost enrollment, and improve trial outcomes. And I think you will see some examples in the next few minutes. Again, thank you so much for the invitation and looking forward for the next presentations.
Thanks very much, Enrique. I appreciate that presentation. Did a wonderful job of overviewing the opportunities and the future state of clinical trial design and inclusion of biomarker diagnostics and how that can advance again the power of trials and again, looking also at some of the potential risks or limitations that we need to overcome in order to make those trials successful here in Ontario. I guess it's my turn now.
Yes.
What I'll do is shift over and just to my deck. Give me a second. Okay, so what I'll try to do with the time I have is provide an overview of just at a higher level. When we look at all trials in the current trial landscape in Canada, for those that are actively recruiting patients, how the trials go about, or how the trials may be findable by those that are looking for trials, whether they're caregivers, the patients themselves, or fellow researchers looking at identifying trials that are currently active in the research landscape. So where does trial finding begin? All clinical trials are, as best practice, recorded and by regulatory requirements in many cases, registered in clinical trial registries. Familiar to most would be clinicaltrials.gov, supported through the NIH in the US, as well as the WHO's ICTRP in Europe. And I wanted to start with registration because it forms the foundation of the data set upon which all clinical trial finding resources draw from. And why is registration important for interventional trials? It's considered really a scientific, ethical, and moral responsibility, deriving points outlined by the WHO in their information set here. Notably, the Declaration of Helsinki mandates the registration of every clinical trial, mandates that the registration of every clinical trial should occur in a publicly accessible database prior to the recruitment of the first participant, underscoring the ethical responsibility there. And again, the ongoing research, healthcare, and health system decisions that are made and derived from available trial information are informed by the available evidence through the registration process and not influenced by selective or biased reporting that could otherwise be a risk without a cohesive whole registry of those trials and a robust data set containing information on all research that has been ongoing or has been completed. Data checks are part of the registry's process for intake for trials and updates to that information. And as a result, can support quality improvements of the trials themselves by enabling early identification in a public setting of potential research design problems, among other benefits. The identification of clinical trials that are in progress can help identify clinical research gaps. So from a researcher's perspective, sponsor perspective, in identifying unmet research questions, the registry provides an overview of that, of the research in a particular area of interest. Improved awareness of similar trials also helps researchers and their funders avoid unnecessary duplication of efforts, duplication of research, and both at a cost and potentially undue introduction of interventions for patient participants. And it can also promote collaboration amongst the research community through that awareness. Posting of trial progress, results, and links to publications improves research transparency and accountability, really a fundamental objective of registries. And related to this talk's content, awareness of recruiting trials by researchers, clinicians, potential participants facilitates accrual. So when we look at this and the best practice and the expectation, the call for including all trials information or all trials in registries, I think we do need to temper that with an acknowledgement that the reality is that trial registry information is often sparse and incomplete. And as a result, the interpretation or the decisions made and the fulfillment of those opportunities that are listed here can be limited, or their limitations are introduced. I think it would be appropriate with this audience to make the call for investigators and sponsors to ensure that the status information for all their trials are routinely updated in the registries, in the registry information. That can include clinical trial sites, updating the sites as new sites are onboarded, lay summaries, inclusion of lay summaries for patients and caregivers, particularly since clinicaltrials.gov itself has gone to great lengths in its latest iteration to include more patient-facing, a more patient-facing interface. Updating the trial status as the trial continues during the open phase. Posting of results and the summary of results is a common gap in the trial registry data. Certainly an obligation to patients and participants and the public in understanding the positive and the negative outcomes of trials is valuable. And as well as any links to publications and findings resulting from that trial, again, should be included in the registry information. Okay. There is an opportunity, I would also point out, in Canada for strengthening our regulatory guidance and a call for Canadian sponsors to really and investigators to create a best-in-class situation for Canada and Canadian trials registry information to be complete. We could strengthen that again through improved guidances, compelling more sponsors to update that information more frequently as an example of how that can be brought about, but a reliance and an improved activity by sponsors and investigators can fulfill that as well. So if we look at our trial finding landscape here in Canada, there are no shortage of resources available for patients and providers to identify active trials. Each, though, however, come with their own limitations. So if we go coast to coast, what I've taken the time to do here is show some trial finding tools that are available in regionally across the country, noting that these trial finding tools start with and have in common the fact that they data set on which they complete trial matching lists is derived from the registry data. So that underscores the importance and the point I was making earlier about ensuring that the registry information is complete. On the back of that, trial finding tools and resources tend to be supported by a back-end process wherein largely manually driven processes where the information, the sparse information that's available on clinicaltrials.gov and other registry data is verified, topped up or enhanced as a result of local knowledge, perhaps about site information. Some finders add more information that's patient-facing, lay summary information, so forth, to enhance and augment the, not augment, but enhance the limited data set that you find in the registries. So they too have their own limitations in how well they do that, but also in how often the currency of the information, how often that data is updated, and the accuracy of the information in those trial finders is dependent on individual processes that support these tools individually. So you see what you have as a result is and what's observed is a varying results based on the use of these tools. They're all fulfill their function, but you will see variations, and it's common to see variations in searching for the same criteria across different tools. Many of the regional tools or tools that are supported by specific groups look at cancer types or trials for specific cancer types are those only available in certain regions. So again, they may have built-in limitations because of that. Awareness of these different tools amongst, and something that Enrique touched on a little bit, is just patients, providers may have limited awareness of these resources. They may have scarce time to search for them. But then there's the question of if you're aware of several resources, the validity of each that can come as well. Adding to that, you've got sponsor and site-specific search tools, patient group website links over and above the examples I showed you there. So 3CTN comes in, and why are we involved, or what's our interest in the trial finding space? It's tied directly to our organization's objectives and activities. We have a key objective for 3CTN over our 10-year history has been to improve equitable patient access, recruitment, and performance to the portfolio of Canadian-led academic cancer clinical trials. Trial finding enables that all of those objectives. We also seek to promote the adoption of EDI best practices in clinical trials and ensuring that every patient, regardless of where they live, has access to trials. And that starts with understanding what their trial opportunities are locally or elsewhere in Canada. And on the latter note, we have in 3CTN led the development of the Canadian Remote Access Framework for Clinical Trials, or CRAFT, which is a decentralized trial delivery platform that can be applied really for most trials, wherein activities or the entirety of the trial activity can be delegated from one site to another site based on that satellite site's capabilities and trial experience and resources. So if you want to learn more about 3CTN and the CRAFT framework, there's a little QR code there. We'll also include and share this slide for you to be able to learn more about those. We also have a foundation of patient partnership and a patient representative community practice that help guide and inform all of the resources that we develop and our strategic priorities. Um, and those resources include two trial finders, related trial finders, two interfaces, trial finding interfaces that we've developed of our own. I also add that to the complicated Canadian landscape. But on the right, you'll see the Canadian Precision Oncology Trial Finder. That is a resource that has been developed by 3CTN. I would say it is an excellent resource in terms of addressing some of the common limitations of other trial finding tools, but also adapting to the precision oncology landscape in a way that enables trial finding with the current roster of PM trials available in Canada. I'm going to provide a quick demo of that to follow, but I'll also mention too that 3CTN has supported the pilot launch several years back of the Clinical Trial Navigator program out of Windsor. Dr. Carolyn Ham leading that initiative, and I'll provide a bit of an overview of that as well. So if we look at the 3CTN Precision Oncology Trial Finder, this is what it looks like if you go to the 3CTN page. All of the visuals on my slide are linked to the actual website resource, so you can click on them when you get the slide deck. But if I do that, you will see, sorry, I'm just going to do that now. Is everyone able to see the trial finder, or are you still seeing the deck?
No, we're still seeing the deck, and
Okay.
I'm gonna note there's about a minute left.
Yeah, it's fine. Okay, thanks, Michelle. Okay, so what I'll do is I'll defer doing that for sharing the demo, but I'd encourage you to click on the link and go through this. It actually allows you to search trials um, by biomarker or drug target, a filter for multiple criteria in that search, and it will turn a detailed list of trials matching that criteria and a link to where those trials are located that's derived from ct.gov registry information. It also includes, as a result of 3CTN's processes, lay summaries and is for all trials and a detailed list of information that isn't available in other trial finders. We also have a systemic process wherein sites routinely report and update on their trial status, so it helps us keep that current. So the Trial Navigator is actually a service and a trial finding service that was launched out of Windsor, as I mentioned. It takes a next step in our precision medicine trial finder that I just showed also has a link to the Trial Navigator for those that would benefit from a supported search. And so the Trial Navigator program has Trial Navigators, personnel, and processes for doing a systematic search for available trials, drawing from available trial finders that I've shown, but also incorporating a clinical review where the matching process can be verified by clinical teams in creating a curated short list of preferred trial fits for a patient. There's a link there to a recent publication from 2022 of the pilot of the program. And the program has since scaled and has supported approximately 400 patients in sourcing available trial information. 24% of those were referred to eligible clinical trials, and 8% of those patients have been successfully enrolled in a trial, which is better than the national average. The process itself, and I'll just wrap here, involves a consent by patients to have the navigator program, and the navigators do the search for trials on their behalf. And that begins with an online consent. Private information is then submitted to the navigator, and launches the search process, the results of which are sent back to the patient and their caregiver to begin the trial discussion, eligibility discussion, can link with the trial navigator to help interpret the results, and the navigator can also support a live connection to transfer to the available site of preference for a given trial. So again, it aims to help take trial finding uh, to I guess the next level, a concierge service to help those that would benefit from it. So I'll wrap there. As I move on to introducing our next speaker, Paul Brogee. Paul Brogee is my next speaker. Paul holds a BSc in molecular biology and a master's in chemical engineering from the University of Waterloo, where his research focused on the use of serine recombinases to engineer, sorry, Paul, to engineer insect cell lines. Now at Princess Margaret Cancer Center, Paul's focused on streamlining and automating the way cancer patients are matched to precision oncology trials through the PMATCH initiative, which is a good segue into your talk.
Thanks, Stephen. So my name, as Stephen mentioned, is Paul Brogee. I am a project manager over at Princess Margaret and working with doctors Benjamin Habey-Keynes and Trevor Pugh on an initiative that they've spearheaded that is aiming to really improve the way that we screen patients, specifically, though not exclusively, for precision oncology trials. And so, as I think both Enrique and Stephen have made quite clear, precision oncology's trials are increasingly complex and involve multiple sites and technologies, and we really need an improved systematic infrastructure for matching patients to these trials. Precision oncology trials in particular are multimodal in nature, and we need genomic data, clinical data, information about the clinical trial design and eligibility criteria in order for both clinicians and trialists to review them and screen patients. And I mentioned both of those groups because we often think about, at least in the PMATCH group, we often think about this system from both perspectives. So sometimes you are a clinician and your patient has reached end of standard of care or has simply progressed to a point where you're interested in looking for trials for that patient. And so you're interested in perusing the many potential trial options for that specific patient, or you may be someone involved in a trial team and wanting to ensure that that trial is available to as many patients as possible, because maximizing accrual to that trial is obviously a crucial component for trial success. That being said, there are a number of roadblocks in order to bring together this multimodal data. In terms of genomic alterations, as Enrique mentioned, there are a limited number of actionable alterations, although that is improving over time. We, in the PMATCH development process, have been talking to many clinical research coordinators and other staff involved in clinical trials in the actual recruitment process for a clinical trial. And they have noted for us time and time again that these very specific genomic criteria are a real pain point. So trying to streamline that process is a real opportunity for us. For clinical data, there's a huge amount of clinical data required to screen a patient, but most of this exists as unstructured clinical notes, which makes it very difficult to work with programmatically. And so there is a need or an opportunity to structure this data in a way that makes it easy to screen using a number of automated systems. Something that Stephen mentioned, the clinical trial information itself, while it is designed to be registered on clinicaltrials.gov, some of this can be out of date, and most of it is, again, unstructured. Even within clinical trial protocols, you have eligibility criteria that is in a free text form that makes it difficult to work with in a programmatic setting, and the same goes for status information. And so the PMATCH initiative is really designed to be a bit of a molecular tumor board in a box, so to speak, where we are tying together a number of software systems that are designed to tackle individual components of these problems, and bring them together in order to increase the efficiency and the matchability by interlinking all of this data together. This diagram is sort of the large scale concept map for how PMATCH works. And I'm going to try to walk through the entire PMATCH system, I think in a very brief period of time, but I'll try to answer any questions people might have in the Q&A section. So we're pulling in trial protocol information as well as that clinical and genomic data, passing that into a matching algorithm, factoring in these predictors of therapeutic response, and packaging that up to clinicians and trialists. And in the future, we have an eye towards being able to make this available directly to patients as well. To start with, we use a software called the CTIMS editor to convert free text clinical trial protocol eligibility criteria information into a machine readable format that we call the clinical trial markup language. This is an extensible markup language that exists as a JSON file and was developed, I believe, at Dana-Farber. However, it's very finicky to work with, if you're not an experienced coder. And so the CTIMS editor is a user interface that has drop-down menus and autocomplete fields and allows you to build a digital, programmatically readable version of a set of eligibility criteria that represents that particular trial. The clinical data that exists at Princess Margaret in EPIC, I mentioned before, exists as unstructured clinical notes. We have a sub-team within PMATCH that is working on developing LLM approaches to be able to structure that data into the MOHCCN clinical data model so that we can work with the specific clinical notes within the PMATCH algorithm. They've been experimenting on a set of 350,000 de-identified notes from a set of 3,400 patients from the Octane group. And those notes consist of imaging reports, cytology, pathology reports, clinical encounter notes, medical history, physicals, procedure notes, etc. It's a really broad set of notes, and our preliminary results are quite promising. So we're hoping to be able to scale this up for other applications. Notably, the trial protocol structuring itself will be our next target to hopefully speed up that CTIMS editor process by involving some LLM work in that workflow as well. The patient genomic data that is so crucial for the precision oncology trials. Right now, we are working with whole genome and transcriptome data that is generated at OICR and then gets passed to a data warehouse CBio portal at Princess Margaret. All of the whole genome and RNA-seq data that gets packaged up with the GIRBA software at OICR, we're able to take that out of CBio portal in a de-identified format and use that to make genomic criteria determinations. So we take all of that information and funnel it all into a matching engine that generates a list of trial matches for each patient, or a list of patient matches for each trial, depending on which perspective you're looking at, and pass that set of matches to a nifty little module we call the prioritizer, which pulls in predictors of therapeutic response, those biomarkers that Enrique did a really excellent job of sort of talking about in detail. I'm going to maybe breeze past some of that because it's been covered in a little bit more detail earlier. But in short, we take the list of matches and compare it to both actionable clinical and preclinical biomarkers. So in cases where there are actionable verified OncoKB biomarkers, we would surface those at the top as for having the strongest level of evidence. In cases where that's not the case, we can use preclinical or experimental biomarkers. For example, Dr. Habe Kane's has a software called PredictIO that is specific for immunotherapy response. And so in this case, we had a patient that matched to 24 separate trials, and were able to use the PredictIO data to suggest that despite there being no OncoKB hits, PredictIO did seem to indicate with some transcriptomic signatures that pembrolizumab would be the best course of action for this patient. And so you could use that to prioritize trials using pembrolizumab. And so we take all of these prioritized results and package these up in a viewing module that I'm going to show a few screenshots from. I want to preface this by saying the data that is going to be shown here is all dummy synthetic data for privacy reasons, but we have a prototype that we are working with some real data and are hoping to start piloting this in the fall. So the hope is that you'll be able to see patients and trials that you're involved in, depending on your sort of individual user authorizations, and can provide some insight as to trends. For example, if you are working on a trial to see how many patients are eligible for a particular trial. We would be looking up at a specific patient. So, for example, we're looking at Octane here, and we're seeing 6 potential patients. We'd be able to click in to, for example, Irving B, and see the specific trials that have been identified by the PMATCH algorithm along with their evidence level. That is suggested by the prioritizer module, and specifically listing how many arms within that trial the patient may be eligible for. So that is really sort of the basic workflow. I think in a few months we'll be showing off a slightly more live demo of this at a 3CTN conference, and I think we're very excited to be doing that. Really, in conclusion, this is a system that is designed to pull in all of the different data required to make some of these screening matches, and provide a pre-screen to ensure that when CRCs are going through the very time-consuming manual process of determining patient eligibility, that they're hitting as few false positives as possible, and decreasing any kind of wasted effort. So that's PMATCH. I'd like to thank a number of the teams involved in this. I think I went a little bit faster than I was anticipating in my practice run through. So I'm done a little bit early, but hopefully that leaves time for some additional questions here.